Hello friends, welcome to BMH Learning. In this video, I am gonna talk about native page. So what is native page? You must have heard about gel electrophoresis, or more specifically, page. That is polyacrylamide gel electrophoresis, a technique which separates proteins under the influence of electric field. So this native page is nothing but a variant of polyacrylamide gel electrophoresis. Now, what is the meaning of native? The literal meaning of native is original. We can say the unmodified form. So, because we are talking about polyacrylamide gel electrophoresis, native here actually deals with original form of proteins. Now, the actual name of gel electrophoresis for protein separation is SDS page. So, you have to remember both SDS and native page are different. Now, it's the SDS which makes it different. We know the original form of protein is folded 3D structure, which is possible because of a number of forces, bonds and interactions happening on the protein molecule. Now, when we treat the protein with SDS, it edits the protein in two ways. The SDS breaks the interactions and forces which are holding the protein in what we call a 3D conformation into a linear form. The second function of SDS is that it imparts negative charge to the protein. Now, in case of native page, no SDS is used. Hence, the protein remains in its original form, and the protein is electrophoresed in the original form itself. So we can say, in native page, the protein is separated on the basis of size, shape, and most important thing, charge. And in case of SDS, this charge factor is nullified. To understand the concept of native page in detail, let's talk about the principle of native page. First, we take a brief look at SDS page procedure. As we know, treatment of protein with SDS makes the protein linearized, and protein comes under the influence of negative charge. Suppose the mixture of the protein to be separated has three types of proteins having different size and molecular weight. As there is only negative charge on all proteins, so it's not a factor for separation anymore. After the electrophoresis is done, the various proteins would be placed at their respective positions in the gel according to their size and molecular weight. But in case of native page, the idea is that, let's say this is the protein, and this protein is in folded 3D conformation. The thing is that the protein has its own, or you can say it has its personal charge. That is, it can be positive, negative, or slightly positive, or slightly negative, any degree of charge. This is one aspect, that is, having a specific type of charge. The second aspect is that we are not breaking it, that is, it's in original form. So let's suppose we establish an electric field here. This side is positive, and the opposite to this one is negative. Let's say we are dealing with three types of native proteins differing in their molecular size, shape, and most important, they are having a different net charge. And remember, that different net charge is coming from specific amino acid sequence a protein is possessing. Now, as we know, they are having different size, shape, and net charge. Depending on this, a specific protein will end up being trapped in a particular location of polyacrylamide gel where it belongs. And every time we do this procedure, we find the protein moved to the same position in the gel. What we can observe and understand from this, we can say how this thing is useful to us. So if we are running native page for, say, protein X, we already know the X protein will migrate up to this specific position. And repeating the procedure over and over again will give us the same result provided we are using the same experimental conditions. So the significance is that The method helps us to know how the protein behaves. It tells about the protein structure as compared to SDS, in which we only want the protein of interest to get separated from the rest. Now let's talk about the application of native page. It's used for the detection of type of amino acid a protein is made of, like hydrophilic, hydrophobic, or we can say positively charged or negatively charged amino acids. It also helps in the detection of protein-protein interactions and to know if a protein can bind to a specific nucleotide binding region. Apart from this, various motif structures can be known through this technique. This was all. Thanks for watching.